The overall goal of this experiment is to provide models of the free radical transformation of biomolecules. The chemical, mechanistic, and analytical information can then be transferred to biology, medicine, and biomarker discovery. This is achieved by first synthesizing appropriately modified biomolecules under free radical conditions initiated by UV light. The synthetic plan can also be designed to simulate free radical processes occurring in a biological environment, which is the principle of biomimetic radical chemistry. As a second step, the products are purified, analyzed, and characterized, which provides structural and chemical information of the modified biomolecules in order to organize molecular libraries and facilitate recognition in the complexity of a biological environment. The protocols used enable the separation of complex mixtures of compounds, such as those derived from biological specimens. Finally, in vitro cell culture experiments and in vivo studies from animals and human are used to develop biomarkers. These results show the importance of chemical biology studies of free radical reactions to the understanding of complex transformations, which can be the basis of signaling and damages in living organisms.
Our group has an interdisciplinary approach linking chemistry and chemical skills to biological processes towards health application. We first study chemical mechanism using appropriately designed biomimetic scenarios for which we developed a library of modification of biologically relevant molecules due to the free radical process. Then we shift to more complex studies in biological samples, confirming the presence of modified compounds as an effect of free radical attack.
Often, free radical stress in living organisms lacks reliable and feasible